So to visualize protein structures, there's a program that you can get out of Apps Anywhere called PyMol. So I'm going to start PyMol. And PyMol starts with a collection of different windows. There's going to be one that allows you to actually display the protein. There's going to be another one which tells you all sorts of console information and text. So it's given me a warning already about something that's missing. The educational version doesn't have all of the functions of the real commercial version that's produced by a company called Schrödinger. So there are limits on the things that we can do. I think you can't output videos, for example, movies of a certain type, for example, which is slightly annoying, but not the end of the world. Right. So I'm going to open one of those PDB files. I'm going to open the one that is easiest to look at, which is going to be the 1L7F. So if I open it, that's what a protein structure looks like. So if I make it full size, I'm looking at it and going, wow, that looks quite nice. You're looking at it and probably going, oh my God, what does that look like? Now this one's slightly more complicated than usual because it's got some other things attached to the protein. All of these red dots mean water. And water is a very important part of protein structures because each of these waters is actually attached to the protein by hydrogen bonds. So when you get to the end of a chain, so here you've got some oxygens. So oxygens are coloured red, nitrogens are coloured blue, carbons are coloured green. So when you get some oxygens at the end, these hydrogen bonds to water molecules. So this is an amino acid with an acidic group on the side, so it attaches itself to two waters that both attach to the different oxygens there. Now, it's a bit difficult looking at this structure and pointing and clicking at things to find out where you are within the structure. So there's another thing we can do. Now, unfortunately for me, I keep having to minimize this so that I can switch on other functions. So what I want to do is display the sequence. So that makes life a lot easier. So here you have the amino acid sequence. So you can see that it starts at position 82. So it should start at position 1. So that means that the first 81 amino acids have been cut off. So you can see it's sequenced in the single letter code. That's all fine. Then you get to the end of the sequence, which is 466. Then you have these other things, which are called GOL, BCZ, CA stands for calcium, and NAG, which stands for a particular sugar. So I've selected those things. So what I can do now is just show those things, or I can hide those things. So when I clicked on them, I got an extra line here which says selection. So what I can do is do some sort of action with the selection, so I can delete that selection, so start again, or I can zoom on it or reorientate the way it rotates around that particular selection and do other things like that. Or I can show that selection only and then delete all the other things, so you just have a focus on that. So if I show those just as lines, if I still got those selected, so if I do hide everything temporarily, I didn't hide everything either. Sometimes oh, that would be better. Let's hide everything. That's hid everything. So that's hidden the whole protein file. Now I want to show as lines those particular things that I've highlighted, the two goals, the BC and whatever. So if I unselect these things now, you can see them as lines. I should be able to select two buttons and zoom it. Yes, I can. So I can zoom in and look at these things. Now, these are why this is called a glycoprotein. It's called a glycoprotein because it attaches to the sugar residues and these other compounds. So the calciums are just there as counter ions, but NAG and MAN, which is mannose, these actually attach to the protein itself. So if I do sticks for, oh, that sticks for everything. Right, so I've gone back to showing my original protein. So you can see that this long thing over here is a sugar group that's attached to the side of the protein. So I can make that easier to see by changing that to having sticks instead of lines. It makes it bolder, and then I can remove the selection. So now you can see that glycosylation there, and there's another glycosylation over here, which I've missed that, selecting that last sugar. So these are mannose molecules, and they can branch out in all sorts of strange shapes. So the proteins on the outside of this virus have sugars attached to the protein itself. That's the process of glycosylation, and they are glycoproteins. That's important because it specifies how the virus gets into your cells. Now, I'm not very interested in that aspect, so I don't really want to look at those particular parts. What I'm more interested in is the protein itself. So what I'm going to do is select the first residue, then go to the end of the protein, press shift and select the last residue. So now I've picked the protein in my selection. So now I'm going to hide everything from the whole molecule, and then I'm going to show as cartoon the protein, and then I'm going to remove the selection so we don't have all those dots. So this is a more usual way of looking at a protein structure. So this is a cartoon representation where each one of these arrows represents a beta sheet, and the alpha helices are these helical swirls. Now if I look from this particular angle, I'm looking down into the center of a beta barrel. So that means this is the center of the protein. This is where things are going to happen. So it's where it's going to attach to the outside of the cell so that the virus can get in. What's special about this one is these helices. These are not present in the bat version. So when I overlap them, they're not there. Looking at it like this, it's not very easy to understand. So it's better to do some colouring. So you can do colouring by each of the different elements, which isn't going to make any sense with the secondary structure. So I want to colour by SS, which is secondary structure. Now I can choose these three different colour schemes. I can have red for helices, yellows for sheets, and green for loops. This is quite a standard way of doing things, and you'll see this often in textbooks. It's nice, I'm used to it, it makes it much easier to understand. Only problem is if you've got somebody who's colorblind, that's not a very good choice of colors. So I would suggest trying one of the others. So this one has a magenta for the sheets, has blue for the helices, and this undefinable sort of pinky colour connecting things together. So we'll leave it like that because that's not causing me any particular problems. So a couple of the projects ask you to identify what the important amino acids are. And a good thing that you could do, especially in the hemagglutinin case, where you're asked to actually demonstrate where they are in the structure, is to identify the important and unique amino acids and then add them to this drawing. So that's what I'm going to do now. So what we have is the neuraminidase alignment from aligning the two proteins. So what I've done is I've opened it in Mega, and what you can see here is the alignment between the two amino acid sequences. So the one that I've got displayed at the minute is this bottom one. All the places that have stars are probably important. Particularly, I highlighted that all these C's, so the C that's here and here and here, and are there any more C's? And here, and here, and here, and I think that's probably it. They're important because they're in every single neuraminidase. Oh, there's one more down here. So that's important. It's also likely to be important. Oh, the W at the end. That's interesting. Also likely to be important where you've got lots of these stars bunched together in groups. So this region of the sequences seems to be doing something important, and this is definitely really, really important. So let's focus on this bit, which we know to be very important. So let's go from this L to the DG that's here. So I go back to my protein here. I've got to find where they are. So I'm looking for the pattern LRTQ, whatever. It's a bit difficult with the different colouring. Actually, what numbers are they? 143. So let's go to something around 143. Nearly 143. LRTQ doesn't look like it's there. Remember, I have to add 80, so it would be about 220 actually. LRTQESES. This is where we are. So I want to highlight LRTQ, not the E because it's an A in the other one. S's are both the same. S and E is not the same. The C is the same. The I is not the same. The C is the same. And then there's a D and a G, which only the G is the same. Then the C is the same. Now I go to that D and G. Right, so I've highlighted all of that region within the protein. So you can see, if I rotate it around, that they all fall along that particular loop region of the protein. So that suggests that this bit here is doing something important. So it's probably involved in the activity of this particular molecule. Now, to make that a bit clearer, what I can do is show those. Whoops. just want it for the selected thing, as sticks. And I can color those by element. What color scheme do I want to use? Let's have orange for carbon, blue for nitrogen, red for oxygen, sort of yellowy color for sulfur. Does that work? Well, vaguely. So I've deleted the selection. So now I can look at it in detail and say, oh, these are the things, the parts of the protein that are always conserved. So what do I think they're doing? Now, something that's interesting here, which I should select and try and zoom on that one. So center on that particular thing. And then I want to also zoom. Zoom, not so much. Can I less zoom? That's a bit of over zoom. That's a bit better. Let's not zoom so much. You also see that what you have is a depth of field, so that to make it less complicated for you, it has a slab that it cuts through the protein to make things visible and not visible. So there is the bit of the protein that seems to be important. And the bit that I wanted to show you is this bit here. So it's two cysteine residues beside each other. What they've done is formed a bond between the two S's, between the two sulfur atoms. So this is called a disulfide bond. And within neuraminidase, there are about 6 or 7 disulfide bonds which are really important. And they're totally conserved through all of the neuraminidases. So they're things that hold this loop to this helix. So it determines the shape of that loop. Right. So to save myself time, here's one I prepared earlier. Oops. So that was the standard. That's the complete molecule with all of the highlighted conserved amino acids that are conserved between both of the protein structures. So you see those in orange. So you see that most of them fall in the center of this beta sheet. So this has to be conserved because it's the middle of the protein. Oh, same thing, different color scheme. That's my better purple one where I'm showing. So you see here, there's another disulfide bridge. There's another disulfide bridge. There's you can see 3 here in the same place. So all of these disulfide bridges are important to the structure of the protein. So evolution cannot change them. These are fixed things that are important for the protein to stay folded together. So the last thing I decided to do was just create an image of those conserved parts of the protein and then get rid of the cartine. So this shows you this central core which has been preserved in evolution. Some other things that I thought were interesting is if you look at, so here you've got 2 ring amino acids. So you've got phenylalanine and a tryptophan. Actually, no, it's a tyrosine and a tryptophan. And the 2 things are stacked on top of each other. So because these are both aromatic compounds, this is called pi stacking. It means their pi bonds are stacked to each other. It's the same as in graphite in chemistry. So by looking at this carefully, you can begin to explain the chemistry of how this thing works and why it folds. So that is the objective of one of the projects. You have to look at the hemagglutinin and identify the bits of hemagglutinin that are important. Now, in neuraminidase, the shape is not very exciting. So as I told you, it's what we call a sort of beta barrel. So all of these beta sheets are arranged around a central core, and there's a little bit of alpha helix on the side. For the hemagglutinin, it's a lot more interesting as a structure, because what you've got is something that looks a bit like a pin. So it has a head at the top of it, and it has a long stalk, which is the thing that fixes it to the outside of the virus. So the thing in COVID, which is very similar to hemagglutinin, is the spike protein. So whenever you see a news report giving an illustration of what COVID looks like, you see a sort of blob, a round sphere with a sort of rough surface, with some spike things stuck on the outside of it. In influenza, the hemagglutinin is the thing that is those spiky growths on the outside. Neuraminidase, this thing, is related in a vague ways to the hemagglutinin. This releases the virus if it gets stuck to the wrong type of cell. So when the virus goes around, it's looking with its hemagglutinin to try and latch on to the outside of your lung cells or a duct's gastrointestinal cells. Now, if it gets stuck in the mucus and it can't get into the cell, neuraminidase cuts it free. That's the job of it. So if we create drugs against neuraminidase, it makes the virus less effective and it can't escape from the cells. So it's a reasonable treatment. But a better treatment is to stop the hemagglutinin, which would prevent the virus ever getting inside your cells. So if we want to make an antiviral drug for COVID, we need to target the spike protein, because that's the thing that's enabling the virus to get into each of your cells. Right. So did you have any particular questions about that? So hopefully that will help the people who have to do some structural